Good morning, everyone. Good morning. Good morning. Good morning, all. I think no one has wake up. Good morning, everyone. Okay, so friends, we are having one more class. We are having one more class at 11:30 a.m. Where I am going to finish the left part of the secondary metabolites as well as the DNA repair mechanisms also. Okay, so please kindly attend even that particular class also. Okay, so friends, so after the DNA repair mechanism in this particular unit 3, we are left with only two topics, that is the prokaryotic gene regulation, then followed by friends eukaryotic gene regulation. Okay, so the two concepts that we have to study in this particular unit 3. Okay, so friends, both are extremely important. If you look into 2022 paper, we got a 4-marker question from the prokaryotic gene regulation also. We got the question from the eukaryotic gene regulation also. So please kindly don't forget to read and to cover both the topics. Okay. So everyone quickly join. Good morning, Srikant, Saima. Oh yeah, good morning all. Okay, so let's start this particular operon. So, good morning, Priya. Okay, so I will explain the operon in a very simple and effective way. By that you can understand everything elaboratively. You know, friends, if you comes to this particular operon, this is 100% concept oriented. 100%. I mean to say there is nothing to read, nothing to remember, 100% to be understood. If you understand the concept clearly, you can answer any kind of a question, right? Good morning all. Good morning, please continue. Okay, what kind of questions we can expect in this particular operon? Friends, operon is the one we can expect almost every kind of a question. We can get the question that is factual statement related. So friends, we can get the question analytical, and especially friends, we can get the questions from the mutations, right? And friends, not only from the mutations, we can get direct experimental based questions. Direct experimental based questions also we can expect from this particular operon, right? So we have to read every single concept in this particular operon. Let's start it. Okay, so friends, I'm requesting all of you before starting, pay utmost attention whatever we are discussing. Okay, first listen to me 20 minutes correctly. I'll make it extremely simple. After 20 minutes I'll solve all your doubts if you have any kind of a doubts. So pay attention. Polycistronic and monocistronic. Okay, we already discussed even in the transcription also. What is polycistronic, friends? In case of prokaryotes, the RNA is only one. One mRNA can synthesize multiple proteins. Is it, friends? One mRNA can synthesize the multiple proteins. So, but what about in case of us? We are very complex organism. Especially, I am talking about the humans. In case of us, friends, one gene, one protein, one gene, one protein. Because we are highly complex organism, one gene cannot code for all the proteins present in our body. But if you look into the prokaryotic organism, their genome is a small genome. Their genome is not a complex genome, friends. As a result, one mRNA can code for the multiple proteins. so which we can call polycistronic. So polycistronic only occurs in the prokaryote organism, whereas eukaryotes basically shows the monocistronic. So friends, listen to me for 10 minutes continuously. I'll make it very simple. A bacterial metabolism, right? So we all people know, right? We are having two type of operons. One we call it as the Lac Operon, another one we can call it as the tryptophan operon. So friends, always every one of you remember Lac Operon is a best example for the Go Operon. Tryptophan operon is the best example for the stop operon. I mean to say, friends, the Lac Operon is just opposite to the tryptophan operon. So friends, every student listen my statements very carefully. All of you listen. I am a bacteria. What friends? I am a bacteria. Inside my body, glucose is there and lactose is also there. So obviously what I will use first? Undoubtedly friends, I will start using the glucose first. Why? What is the reason that I will use only glucose first? Why not the lactose? Because friends, glucose is a very simple sugar. Hence, I do not require, being a bacteria, I do not require any kind of the enzymes to break down that particular glucose. So that is the reason I will always prefer the glucose only. But if glucose is completely depleted, there will be no presence of the glucose. Which is available? Only lactose is available. If I want to use the lactose, what I required? I always required, friends, everyone listen, I always require certain enzymes which can break down the lactose into simple sugars like glucose and galactose. Then only I can consume the lactose. Otherwise, I cannot consume the lactose. Okay, if I want to break down the lactose, what should I do first? I have to synthesize certain enzymes which are required to break down the lactose. So what I have to do? Whenever there is a presence of lactose, I have to switch on the operon. Why I have to switch on the operon, friends? To start the production of the enzymes for the process of digestion. To start the production of enzymes for the process of digestion, we have to turn on the operon. So my friends, always simply remember, listen my statement. Whenever there is a substrate, whenever there is a presence of the substrate, operon will be turned on. What is the substrate here? Of course, lactose. So in a simple way, when lactose is present, friends, operon will be turned on. Now, what about the tryptophan operon, friends? Tryptophan operon will be completely opposite. What is the speciality of this particular tryptophan operon, friends? All the students try to listen to me very carefully. Tryptophan operon, whenever there is a tryptophan is present, the operon will be turned off. It is completely opposite when compared to the Lac Operon. All the students don't get confused, friends. Try to understand what exactly I am saying. I am a bacteria. In my body, what I will do? Every single time in my body, I will be starting synthesizing the tryptophan. There is no doubt about it, because what is tryptophan? Tryptophan is an amino acid, right? So amino acid has to be synthesized in my body continuously. But what you have done? You have provided me the tryptophan externally in the form of a injection, in the form of a medium. I'm so sorry. You provided me the tryptophan in the form of a medium. Then obviously what I will do? Whatever the tryptophan that you have provided me externally in the medium, obviously I will take that tryptophan only. Okay. When I take the tryptophan provided by you externally, but inside my body also tryptophan is synthesizing already. So what should I do now? I have to stop the production of the enzymes for the biosynthesis of the tryptophan. Please kindly do not forget the point. What I would like to conclude in one line. If there is a presence of lactose, Lac Operon will be turned on. If there is a presence of tryptophan, there will be a operon will be turned off in case of a tryptophan operon. Please kindly do not forget that point. So friends, there are three type of genes are there. After this we will understand the operons very clearly. So let me explain you friends, 20 minutes continuously, you will get a very good idea. You will understand the concept very clearly, and this PPT will help you 100%. You no need to read any other book and any other matter also. This will help you 100%. Try to understand it correctly. So friends, there are three type of genes are there. You can see here, one we call constitutive genes, inducible genes, repressible genes. Everyone pay attention. So friends, what are constitutive genes? Everyone try to understand. Constitutive genes are those type of genes, friends, which are expressed all the time. So continuously expressed genes, we can call it as constitutive genes. You can take an example of metabolic genes, whether we are working, whether we are not working, whether we are sleeping, whether we are not sleeping. So our metabolism should be active. Certain times metabolism will become low, but the metabolism is active all the time. So that metabolic genes basically acting as a constitutive genes, they will be expressed all the time. And you can take another best example, ribosomes. Ribosomes are needed constantly for the synthesis of proteins, which means friends, the ribosome coding genes will be active all the time. So those kind of genes only, friends, we will be calling it as the constitutive genes. Next friends, inducible genes. I want every one of you to understand very clearly. Inducible genes can be active whenever there is a presence of inducer. Simple point. Inducible genes will be active presence of inducer. Same way friends, repressible genes will be active by the presence of the repressor. Such a simple point friends. Inducible genes are getting activated in the presence of inducer. Repressible genes are getting activated in the presence of the repressor. So the best example for the inducible operon is the Lac Operon, and sorry, the best example for the repressible operon is the tryptophan operon. Very easy friends. Now from here every one of you pay attention. I'll make it very simple. You will understand it very clearly. So how can we define the word operon? All the students listen the definition what I will give. Operon means a group of genes. What friends operon means? Operon means group of genes or cluster of genes. Operon is a group or a cluster of genes which are under the control of a single promoter. What friends? Operon is a group of genes or a cluster of genes which are under the control of a particular promoter. Now friends, everyone try to understand the concept very clearly. What is the organization of the bacterial operon? What does the operon consist of? I want every one of you pay attention towards this particular diagram. Listen to me carefully for 15 minutes. It contains structural genes. We will read what kind of a structural genes. We already know that in case of Lac Operon we are having Lac ZYA genes. In case of tryptophan operon we are having ABCDE genes. Now after the structural gene there is a presence of the operator and promoter. My friends, I want every student has to remember one important point. What is that? 99% friends, operator and promoter like this, they will be overlapped with each other. See here, they are joined, they are overlapping with each other. 99% they will be arranged like this. If not, friends, operator and promoter, they will be, all the students listen, operator and promoter located very close proximity, which means, friends, they will be associated very close with each other. So 99% either overlapping with each other or they are associated very close to each other. And friends, you all people commonly know that. Anyway, always remember, when you heard the name of a operator, the common point has to come in your mind. Operator is the binding site for the repressor protein. Very simple point. Promoter is the binding site for the RNA polymerase enzyme. Simple point, friends. Okay, now when you look into the regulatory gene is what, friends? It is basically regulatory gene is the one which synthesizes the repressor protein. That repressor protein only, friends, will come and associated with this particular operator. Please kindly do not forget the statement at all. What, friends? Let me quickly summarize you. There are structural genes followed by the operator, where operator is acting as the binding site for the repressor. There is a presence of promoter which is acting as a binding site for the RNA polymerase enzyme, then followed by a regulatory gene which is basically synthesizing the repressor protein. That repressor protein will come and associated with the operator. Don't forget that points at all. Okay, now let's start discussing about the Lac Operon. Lac Operon means, friends, what? Lactose operon. So, Lac Operon is a type of operon or a group of genes with a single promoter. Because, friends, what is this all Lac Operon about? All these structural genes are going to code for enzymes. Those enzymes are involved in the breakdown of the lactose into glucose and galactose. So in a very simple word, I can tell to all of you, the operon will be turned on only in the presence of the lactose. Operon will be turned off in the absence of lactose. See, we have to read a very depth of the concept. So every one of you pay attention towards this particular slide. This slide is at most important slide. Very, very, very important. 90% of the concept is completely relied on this particular slide only. If you understand this slide correctly, the upcoming slides will become very, very easy to all of you. Okay, so let's take the conditions. What are the conditions we will take? What happens in the presence of lactose? What happens in the absence of the lactose? So all of you look into this. These are all what, friends? These are all the different type of the structural genes. The structural genes are what? LacZ, LacY, LacA, operator, promoter, cap site. We will explain you what is this particular cap site also. And friends, here what gene is present? Regulatory gene is present. What is the regulatory gene? That is the one which is basically synthesizing the repressor. Everyone pay attention, friends. Now let me tell you I already told you, right? Listen to me carefully. We discussed that operator is the binding site for the repressor. Promoter is the binding site for the RNA polymerase enzyme. So please listen to me very carefully, friends. Let's consider the condition there is a no lactose. I mean to say, in the absence of lactose. Everyone listen to me very carefully, my friends. If there is a absence of lactose, is there listen to me, in the absence of lactose, is there any necessity to turn on LacZ, LacY, LacA genes? Not at all. We do not require it at all. Because without lactose, if I synthesize the enzymes, they will be inactive because they don't have any function. So friends, in the absence of lactose, we have to block the synthesis of all these enzymes. How can we block the production, or how can we block the synthesis? With the help of a repressor. Every student listen to me carefully, friends. In the absence of lactose, the regulatory gene will synthesize the repressor, and friends, the repressor will come and associated with what? Operator. The movement repressor will come and associated with the operator, friends, operator will be completely blocked. So friends, whenever there is a blockage of the operator, immediately what is going to happen, friends? The RNA polymerase come and attaches to the promoter. But do you think RNA polymerase enzyme can cross the operator? Not at all. RNA polymerase can't cross the operator, then there will be no production of the lac Z, lac Y, lac A. Simple point it is. Now, imagine a condition, there is a presence of lactose. So imagine the red color, whatever I am drawing, this is only lactose. We will discuss that. The isomer of the lactose, which we call it, friends, allolactose. Whenever there is a presence of allolactose, when the lactose, so there is a presence of lactose, obviously we have to synthesize the 3 structural genes, compulsory, friends. Now every student listen to me. If we want to synthesize the 3 structural genes compulsory, what we have to do, friends? So the repressor should be blocked. Once the repressor should be blocked, repressor come out of the operator. Simple point, na? Repressor come out of the operator. When the repressor come out of the operator, friends, RNA polymerase enzyme will go and attaches to the promoter. It can easily cross the operator. Then there will be a synthesis of lac Z, lac Y, lac A. Absolutely right. You all understand this mechanism. You are reading from the +12 level also. But friends, here comes an interesting point. In the presence of lactose, RNA polymerase enzyme attaches to the promoter, cross the operator because operator is free, and synthesize the 3 structural genes. But my friends, the question is here, even though RNA polymerase enzyme synthesize the 3 structural genes, friends, it is not an efficient transcription. Once again, listen to me. It is not an efficient transcription. So the transcription will occur at the minimal rate only. My friends, every student has to remember, if the transcription wants to occur 100%. What, friends? If the transcription wants to occur 100%, every student listen. RNA polymerase enzyme has to get a positive signal. What, friends? RNA polymerase enzyme has to get a positive signal. How does RNA polymerase is going to get the positive signal? With the help of a proteins called as a cap and cyclic AMP complex. This is at most important point every student has to remember. If you understand this, all the cases are extremely easy, extremely. So whenever there is a presence of the cap and cyclic AMP complex, all the students listen to me very carefully. So this is the cyclic AMP, we all know, right? Listen to me. Powerful secondary messenger. The cyclic AMP activate the cap protein. The activated cap and cyclic AMP, friends, they will come and attaches to which site? They will come and attaches to the cap site. Once they come and attaches to the cap site, here what is coming, friends? RNA polymerase enzyme is coming. RNA polymerase enzyme having the C-terminal domain. So friends, this cap protein will give the positive signal. Listen to me carefully. This cap protein will give the positive signal to the C-terminal domain by the process of phosphorylation. It will just add the phosphate groups to the C-terminal domain of the RNA polymerase enzyme. As a result, friends, RNA polymerase enzyme will become active. Once RNA polymerase enzyme become completely active and lactose is also present, then friends, it can cross the operator and it can synthesize the structural genes very effectively. So friends, let me tell you the final statement. What is the final statement? If 100% of the active transcription has to occur, two situations should happen. What is the first situation? That is presence of lactose. That is presence of lactose. In the presence of lactose, repressor will become inactive. We all know that. Second one, the presence of cyclic AMP and cap complex. Friends, whenever there is a two conditions are fulfilling, then only friends transcription will happen 100%. So my friends, the last and final point every student has to understand, when the cyclic AMP will be active in the cell. This is the unique point among all. Cyclic AMP active in the cell when the glucose level is completely low or glucose level is completely absent. This is the unique statement. When the glucose level is low or completely absent, then the cyclic AMP will be active. Cyclic AMP will go and activate the cap protein. My friends, then cap protein give the positive signal to the RNA polymerase enzyme. And one more is what? There is a presence of lactose. As a result, repressor will come down, operator will become free. Then only friends, the transcription occur more efficiently. I hope everyone understand this slide. Now you will only can answer all these very easily. Okay, I want every one of you look into this and pay utmost attention towards this. Okay, so let me explain you friends the four possible situation. I want every one of you to understand. If you understand this slide and this slide, 60% of the questions will come from this only. If you miss the questions from here, there is only one way, that is the mutations. Okay, so first we will cover this, immediately we will cover the mutations. So please try to understand. If you look into this 2022 question paper, we got the questions from the mutations, and that too very easy question. We will solve it. Now try to understand the four possible situations. All the students has to understand. When the glucose is present, lactose is absent. See friends, you only try to understand lactose is absent. Very simple. When there is an absence of lactose, is there any necessity to produce this particular beta galactosidase enzyme? No, there is no necessity at all. Why we have to produce? There is no lactose, then what we will break? Hence friends, there is no need to produce the beta galactosidase. Now look into the second condition, a kind of a logical condition friends. Glucose is present friends and lactose is also present. Kindly don't confuse. Glucose is present, lactose is present. Still there will be no production of the beta galactosidase. Why? Friends, glucose is present. Then what we will always use, I am a bacteria. When there is a presence of both glucose and lactose, undoubtedly 1000% I will always use the glucose. I never go for the lactose, because lactose is a disaccharide. I require lot of enzymes. I require lot of enzymes to break down the lactose. So that's why I don't go for the lactose. I will start utilizing the most simple sugar, that is the glucose. Hence friends, there will be no need to produce the beta galactosidase even in the second case. All the students listen to me, the third case. When glucose is absent and lactose is also absent. Friends, lactose is absent. The point is very clear. When there is no lactose, what I have to break down? I can't break down anything. But then why I have to synthesize the beta galactosidase enzyme? Obviously, I won't produce the beta galactosidase enzyme. Now, the fourth and the most important condition, friends. What is that fourth and important condition? Whenever there is a absence of glucose. Once again, every student listen to me. Whenever there is an absence of glucose, I have already told to all of you, you will see in the next slide. When there is an absence of glucose, what will be active? Cyclic AMP will be active. You will understand. Whenever there is an activation of the cyclic AMP, which will be active, my friends, cap protein also will active. And what did I tell you, friends? Cyclic AMP and cap protein is going to give the positive signal to the RNA polymerase enzyme. And what is the favorable condition? Lactose is also present. What happens now? When the lactose is present, we seen in the diagram, lactose will go and associated with the repressor. Repressor fall down from the operator. Then RNA polymerase enzyme can easily cross the operator and it can synthesize the beta galactosidase and all other structural genes. Then friends, operon will be turned on. Very simple. If you people understand this particular slide, I told you right, 60% of the questions you can easily answer. So everyone pay utmost attention. Let's try to understand the possible condition. Friends, this is really important slide. Very, very, very important. you people no need to remember anything. If you understand correctly, friends, you can answer any kind of a question. So listen to me, friends. We are understanding what kind of sugars are present. What is the status of the cap activator protein, which we can call the cap protein. What is the status of the repressor protein? What is the status of the RNA polymerase enzyme? What is the status of Lac Operon? I will explain this briefly, friends. If anyone having any doubt, please ask me immediately. I will explain to all of you once again. This is most important and easy to understand if you apply the brain. Let me explain. First condition, glucose is present. Lactose is also present. So friends, the concept is very clear. Since there is a presence of glucose, do you think cap protein will be active, my friends? Not at all. Cap protein is not going to be active at all. What is the reason? Because in the presence of glucose, cyclic AMP will be inactive. When the cyclic AMP will be inactive, my friends, cap protein is also inactive. Very simple point it is. When the cap protein is inactive, they never ever associated with the promoter, near the promoter. Okay, now look into the condition, my friends. Listen to me carefully. What is present? Lactose is present. When the lactose is present, lactose will go and associated with what? Repressor. Friends, whenever the lactose will go and associated with the repressor, what happens? Repressor will come out of the operator. What, my friends? Repressor will come out of this particular operator site. Then immediately what happens? Do you think RNA polymerase enzyme will associated with the promoter very tightly? No. What is the, you will get one doubt. Sir, here operator is free. That is true point. But friends, RNA polymerase enzyme, when it associated with the promoter, it keeps falling. Why? I can tell you, friends, RNA polymerase enzyme is not getting the positive signal. Why the RNA polymerase enzyme is not getting the positive signal, friends? The question is very clear. There is a no cap protein. Why the cap protein was not there? Because there is a presence of glucose. Hence, no transcription. So now look into the second condition. Glucose is present, lactose is absent. You will only can understand easily, friends. Same, right? Glucose is present. When the glucose is present, do you think cyclic AMP will be active? No. Do you think if the cyclic AMP is low, cap protein is inactive, then they are not bound to the DNA near the promoter? Not at all. Now look into the condition, absence of lactose. If there is a no lactose, which will be active, friends? Undoubtedly, friends, repressor will be active. We all people know that. When the repressor will be active, where the repressor will go and attaches repressor will go and attaches to the operator. The moment repressor will go and attaches to the operator, operator will be completely blocked. Do you think RNA polymerase enzyme can cross the operator? Not at all. Hence, my friends, there will be no transcription at all. Now, third and fourth conditions are little confused condition, but little logical. If you find the logic, friends, you can easily understand it. Listen to me carefully. There is a absence of glucose. There is a absence of lactose also. My friends, whenever there is a absence of glucose, you only keep on answering, friends, no glucose. No glucose means we already read, right? Cyclic AMP is present. When there is a cyclic AMP is present, cap also present. Good point. Now they will come and bound to the DNA near the promoter and they are ready to give the positive signal to the RNA polymerase enzyme. They have given it also. But the point is here, now RNA polymerase enzyme got the positive signal and it is ready to synthesize the structural genes. But if it wants to synthesize the structural genes, it has to cross the operator. Yes or no? Of course, yes. It has to cross the operator. But friends, if it wants to cross the operator, operator should be free. But do you think operator is free now? Not at all. Because friends, lactose is absent. When the lactose is absent, repressor will be active. Now the repressor will bound to the operator. Then RNA polymerase cannot cross it. Hence there is no transcription. The only way or the only possibility when the transcription will be 100%. Every student has to listen this point. The transcription will be 100% possible whenever there is a absence of glucose and whenever there is a presence of lactose. That's all. This is the only condition that is possible. Listen to me. Whenever there is an absence of glucose, which concentration become high? Cyclic AMP concentration become high. When the cyclic AMP concentration become high, the cap protein concentration also become high. Then what is going to happen? So the cap protein will come and attaches to the DNA. Now it will give the positive signal. Now RNA polymerase is ready to do the efficient transcription. But if the RNA polymerase want to do the efficient transcription, lactose should be present. But my friends, there is a presence of lactose. There is no doubt about it. Since there is a presence of lactose, my friends, what's happening? The lactose will go and associated with the repressor. Then my friends, repressor will be lifted off from the operator site. Then when there is a no repressor, operator become free. So what is the two condition? RNA polymerase got the positive signal, operator is also free. Then RNA polymerase enzyme cross the operator. Then it will do the efficient transcription of 100%. So my friends, what is the statement I would like to give here? The statement I would like to give here is that if 100% of the transcription wants to occur, there should be absence of glucose, there should be presence of lactose. And this is one condition. Another condition, there should be presence of the cyclic AMP and a cap protein. There should be repressor block the operator. I'm so sorry. A repressor should be inactive. I'm so sorry. Once the repressor will be inactive, operator will become free. When the operator become free, RNA polymerase enzyme can easily cross the operator in the presence of a cap protein. When the cap protein will be present, when there is a no glucose. So this is the 4 possible condition. So I told you na 60% of the questions in the question paper will come from here only. Okay. Is it clear everyone, friends? Till this slide I am requesting all of you, most of the people will get some doubts. I am requesting all of you to ask me the doubts whatever you have from this particular chart. Let me know, friends, let me know. Is that clear? I am giving you the 100% assurance. The question won't go beyond the level whatever we will discuss, whatever we are discussing in this particular PPT also. Friends, please give me the answers. Is that clear for everyone? Please respond, please respond. Okay, what about others? Okay, perfect, perfect. Please let me know if anyone have any doubt. Great. Okay, so friends, 60% of the questions will come from here. That's good. Now remaining 40% questions, what is the chance that we can expect? There is a chance that we can expect the questions from mutations. I will explain you the mutations also. Trust me, without reading anything, you can answer the questions very directly. Okay. Listen to me very carefully, friends. Focus. This part is also extremely important part, my friends. Promoter, operate. What does the operon contains? Promoter, operator, structural genes. We have already discussed, friends. There are 3 types of structural genes are there. One we call LAC-Z, LAC-Y, LAC-A. See, most of the students will think that, anyway, what is the function of LAC-Z beta galactosidase? It is the one break down the lactose into glucose and galactose. 90% of the students will think that LAC-Z is the most important one. There is no doubt about it. But friends, when compared to the LAC-Z, LAC-Y is more important, because friends, when can we break down the lactose into glucose and galactose? Whenever lactose enter in our body. That is the important point. If the lactose is not entered in our body, how can we break down the lactose into glucose and galactose? We cannot do this, right? So the statement I want you people to remember here is that whenever the LAC-Y, what do we can call? Permease. What the enzyme permease will do? Which will allow the lactose to enter inside. Which means, friends, it will increase the permeability of lactose. Whenever lactose enter inside, then only everything is happening. So that's why, friends, when compared to the LAC-Z, LAC-Y is more important. LAC-A, trans-acetylase enzyme. Friends, till today we did not understand the exact role of the trans-acetylase enzyme. But many books and many papers suggesting that it may be extremely responsible for stability of the operon. What, my friends? It may be extremely responsible for stability of the operon. Very important point it is. Now, friends, if you understand this slide and if you understand this slide, 90% of the mutation related questions you people can answer very easily if you really understand these 2 slides. Okay, let me explain you. I want you people to listen to me very carefully. 2 points. What is the first point? Operator. All the students listen to me. Operator is the integral part of operon. Always remember these 2 statements. If you listen to me correctly, trust me, the mutation part is very easy. Operator is the integral part of operon, whereas repressor is not integral part of the operon. Never ever forget these 2 lines. Okay, let me explain you. What do you mean by the word operators are cis-acting? What do you mean by the word repressors are trans-acting? Pay utmost attention. Both are same genes containing LAC-Z, LAC-Y, LAC-A, operator, promoter, regulatory gene. Every student listen to me, same gene. regulatory gene, promoter, operator, Lac Z, Lac Y, Lac A, all this. Friends, every one of you pay attention. This is operator, right? What is operator here? Operator is the integral part of this gene. I mean to say, operator is not arranged separately, right? Operator is arranged within the gene. Listen my statements carefully. Operator is arranged within the gene. Never ever forget. So there are two in this gene, one operator is present. In this gene, another operator is present. Every one of you has to listen to me very carefully. Now this operator got mutated. Use the brain, apply the knowledge. This operator got mutated. My friends, do you think when this operator got mutated, do you think this operator will come and replace the function of this operator in this gene? It's not possible. You can ask me why. I already told you this operator is the integral part. It is already immersed in the gene. Now this operator cannot come out of the gene. Listen my statements. This operator cannot come out of the gene and cannot replace this particular mutated operator present on the another gene. That's not possible. That's why we can call the operators are what acting, my friends? Cis acting. Whomever did not understand, I will be re-explaining. Every one of you pay attention. Please look into this. For understanding the mutation, these slides are at most important. Operators are cis acting means what, friends? Every operator present in the gene is an integral part. So friends, this operator cannot replace the function of this operator if it's get mutated. The same way, vice versa. The reason the operator is associated within the gene, it cannot come out. If it is freely available, it can easily replace this function, but it cannot. That's why we can call operators are cis acting. But friends, what is opposite here? Opposite is what? Repressors or trans acting. This is really interesting. Same friends, Lac Z, Lac Y, Lac A, all the structural genes, operator, promoter, regulatory gene. My friends, now who is mutated? And anyway, first of all, every student has to remember, does the repressor is present within the gene? I hope you are getting me correctly. My point is, does the repressor, this repressor, does it is an integral part of the gene like a promoter, operator, Lac Z, Y, A? No. Repressor is the one synthesized by the regulatory gene. It is in the free form. Where it will go and attaches? It will go and attaches to the this operator also it can attaches. Even friends, it will attaches to the this operator also. Because friends, it is freely present. It is not associated or bounded in the gene. No. Hence it is it is present in the free form. Wherever the operator is present, it will go and associated over there. Simple point it is. So that's why friends, I can say repressors are the trans acting, which means, my friends, all the students listen, this regulatory gene is working very effectively. This regulatory gene is not working. Hence, my friends, this regulatory gene cannot synthesize the repressor protein. That's true point. Now, but this regulatory gene synthesizes the repressor, no doubt about it. But my friends, this repressor not only associated with this operator, this repressor can also associated with this operator because repressor is free. It is not an integral part. So it can easily go and attaches to the operators wherever the operators are present. But friends, operators are not like that. If one operator get mutated, this operator function cannot be replaced by the another operator. That is extremely important, friends. So that's why we can call operators are cis acting, operators are trans acting. Kindly don't forget the statements at all. Okay. So friends, there is a very little part of the Lac Operon is left. It will take another 15-20 minutes. We will finish it in the tomorrow class. Tomorrow we will finish the Lac Operon also and the left part of the tryptophan operon also completely. Okay. And I also told you friends, today 11:30 a.m. we have the another class. We are left with a little part of the secondary metabolites. I will finish that little part of the secondary metabolites. In the same class, we are left with a little part of the DNA repair mechanisms also. That also we will try to finish it off. Okay. I'm requesting you friends, please attend without fail. Okay. Okay friends, that's all for now. You can ask me if you have any kind of a doubt so far. Is it clear? Hope it's clear for everyone. You can ask me any kind of a doubt. Okay friends, I will meet you at 11:30 a.m. class and I will upload this PPT in the website around 11 o'clock. You can download it. Okay friends, that's all for now. Happy Ugadi to all of you. Thank you so much. Thank you all. Bye-bye. Thank you all.